Okay. Hi, everyone. I'm excited to have the chance to interview the 3 laureates of the Wolf Prize for 2021. Professor Joan Steitz from Yale University, Professor Lynne Maquat from the University of Rochester, and Professor Adrian Krainer from Cold Spring Harbor Laboratory. All made groundbreaking discoveries in the RNA world. A world that was not known until recently, and was known years ago by biochemists and molecular biologists. The COVID RNA vaccine made the RNA field very famous. The RNA now is viewed as a magic bullet. Professor Joan Steitz made fundamental seminal discoveries in the RNA world, from translation initiation site of prokaryotic mRNA already in 1969. When she moved to the eukaryotes, was the first to show the function of small nucleolar RNA known as SNRPs in splicing, which until her discovery were abundant small RNA with no function, no known function. Her discoveries were fundamental, understanding how splicing and what is splicing, removal of RNA that is not needed for the coding capacity of a messenger RNA. Joan contributed to our understanding of how ribosomes are being made, what controls the stability of mRNA and other RNA modification, including the pseudouridine modification that exists in the COVID vaccine and enables its efficient translation. In last year, she studied a very important regulatory non-coding RNA known as microRNA, link RNA. Joan's work is the textbook of the RNA world. And Lynne, Professor Lynne Maquat, discovered the process known as nonsense-mediated decay and showed the world that the cell knows to recognize an mRNA that has a mistake and removes it before it can harm the cell by making a toxic product. NMD, nonsense-mediated decay, functions in 1/3 of human inherited disorders and also important in cancer. Lynne showed that NMD exists in beta thalassemia, and the first clinical sample she found was in Jerusalem in Hadassah Hospital. She then discovered the exon junction complex that marks the position where this important splicing takes place. Professor Adrian Krainer studies the secret of how splicing works and the role of proteins that are essential for splicing, both under normal conditions and in cancer and neuronal diseases, and found the treatment to a genetic disease, the spinal muscle atrophy, or SMA. He studied the genes SMN1 and 2, in fact, a factor that is needed for SNRP assembly, the same SNRPs discovered by Steitz, and was able, using antisense RNA, to rescue and produce the protein in sick children. His drug injected to sick children in wheelchairs made them start walking. His treatment was the first successful RNA therapy. This treatment, known as Spinraza, was approved by many countries and is given in Israel by the National Health Insurance. On a special note, I know the 3 scientists laureates for more than 30 years. I entered the RNA field when it was still small and not famous. I had the pleasure to host in Israel already several times the 3 laureates in the last 2 decades. Now that I told you all about their fundamental discovery, I want to take you to a journey from the past to present and future in your scientific life and your own journey in this fascinating RNA world. The first question I would like to address to all of you is Eureka. I'm sure you had felt it several times during your career. Can you tell us about this moment and what followed it? First, Professor Steitz. Joan.
Thank you very much, Shula, for a lovely introduction. Eureka moments. They're the things that are very, very special in science, where you realize that you've done an experiment and it tells us something that nobody else in the world knows. And you hope that your colleagues are all going to appreciate it, but you're the one enjoying it at the moment. And for me, I would say that's happened twice. What Shula didn't tell you about RNA, or didn't emphasize, is that whereas DNA is double-stranded, and you know that the two strands base pair together, RNA is mostly single-stranded. But that means it has the potential for base pairing with other RNAs. And that's what it does in all these regulatory RNA molecules that we've been talking about. So the first time that I had one of these aha moments was when we had done an experiment that we think actually was the first biochemical evidence for the fact that the way ribosomes, the protein synthesis machines, know where to start on a messenger RNA is by doing base pairing between the ribosome and the RNA of the ribosome and the RNA of the messenger RNA. And the second time had to do with the SNRPs that you've already heard about, when we realized that maybe we only knew a few sequences at that time that were splice sites in messenger RNAs, but we realized that maybe there was base pairing between the RNA in one of these SNRPs and the messenger RNA. So those are my Eureka moments.
Thank you very much, Joan. And can you tell us what you did when you felt it? I mean, what did you do next? I mean,
what did I do next? Well,
yeah.
in the first case, it turns out that there had been a lot of speculation in the field. So having some sort of biochemical evidence was helpful. Later, there was also genetic evidence coming from other labs. And so that was sort of, you know, we finally proven it. In the case of the SNRPs, some people in the field said, wow, that's a great idea. But many, many people were extremely skeptical. There was even a little editorial in Science magazine saying the gloss is off the small nuclear RNAs, meaning that, you know, they don't do this. And for several years, it was sort of a struggle, but we persevered and other people persevered, and eventually the evidence came out.
Thank you very much.
Goes.
So Lynne, tell us about your Eureka moments.
Okay. Well, I study largely messenger RNA, mRNA, the RNA that is the template for protein synthesis in our cells. And mRNA is never naked in the cell. It's always associated with proteins, and we're still trying to figure out what some of these proteins do. It turns out that mRNA is synthesized in the nucleus from our DNA, but it encodes proteins in a different part of the cell, in the cytoplasm. And so our Eureka moment, and we've had several, but this one stands out in my mind right now, is discovering that the first rounds of protein synthesis in the cytoplasm use newly synthesized mRNAs that haven't yet lost proteins they acquired in the nucleus. And these are proteins that were used by the cell to generate the mRNA. We called this new first round of making protein the pioneer round of protein synthesis, the pioneer round of translation. And it turns out this is the round that cells use as a quality control process. And the idea is if an mRNA is going to produce a truncated protein, then the mRNA will be degraded. So the protein won't accumulate. The protein can be either non-functional or even worse, toxic to cells.
So what did you do when you had this idea?
You mean besides jumping up and down and, you know, telling the postdoc in the lab that we would call it the pioneer round of translation? So, you know, it wasn't believed all that as much as I would have liked in the beginning. Yeah. And we had to do a lot of follow-up work. And I think what really nailed and convinced people was work that we did with Rob Singer to actually visualize the mRNA in cells and see where the mRNA was being degraded during this round of translation. And even Rob wondered whether we were right, despite all that we had done over the years. But yeah, it was very satisfying and relieving.
Great. And what about you, Adrian?
Hi, Shula, and thanks for your question. I can think all the way back to very early in my career when I was a student, and I was trying to set up a cell-free system to study RNA splicing in the test tube. And so at some point the conditions were right, and we could see on a gel that there's the band. That means that not only it worked, but it worked really well. And then there were extra bands. So often these Eureka moments are, wait a minute, what's that? And then you realize that if you investigate that, you're going to find something new. So it's a series of steps. And so that extra band was pointing to us that splicing happens in 2 steps. And we didn't know what that intermediate was, but then characterizing that gave us a big surprise, the lariat intermediate. And then much later in my career is when we were trying to develop an antisense oligonucleotide to fix a splicing defect in spinal muscular atrophy that you mentioned. And there, again, there is a series of steps, but at some point we knew we had a molecule that worked and we could treat mice that had an SMA type disease, and they could now live much longer and they could run around instead of being paralyzed and dying in a few days. So that, of course, that was amazing. But then later when clinical trials were being done and we could just follow over a few years, and then I think that's different from a Eureka moment, but it's the same kind of exhilaration, maybe more so, when you realize that, yes, I mean, patients were treated and it worked. I mean, here's the evidence. So in that case, that's a phone call.
Amazing.
As far as, what I did, usually, you get this rush, but then of course you want to tell somebody. So your PhD advisor or everybody else in the lab, or your colleagues, your collaborators, you just want to share this amazing impression that you get.
Oh, you call up Joan. This is what I do.
Would be great.
Yes, so the second question I would like to ask you all. Now RNA is famous, but it was not like that many years. What attracted you to studying RNA in the times it was not in mainstream? Joan, please.
Okay, so for me, it was serendipity and good luck. I was an undergraduate at Antioch College in Ohio, which has a work-study program where half the time they actually send you off to other places in the country or the world to do actual work, and then you come back and the intermediate semesters are on campus. And I was fortunate as a chemistry major to be assigned to a job as a sort of lab assistant in Alex Rich's lab at MIT. And before I arrived there, I hadn't even known, although it was something like 8 or 9 years old, about the double-stranded structure of DNA, but I was sort of interested in genetics. And when I learned about double-stranded DNA, I was just totally enthralled. But the project that I was set to work on was actually an RNA project. It had to do with ribosomes. And although this project was a complete failure, it entranced me by RNA, and the other undergraduate jobs I had and my graduate work was focused on RNA, as was my postdoctoral work. So I was just lucky to have ended up with this particular experience at an early age.
Right. Wow. Lynne, what about you?
I would also say serendipity and luck. I've always studied RNA. So as an undergraduate, I had a lab experience. I took a cell biology course where I learned about protein synthesis and I thought it was so cool. And so I asked the professor, Stu Haywood, if I could work in his lab. I don't know how I knew to ask him. And he said, sure. And so I studied messenger RNA as an undergraduate. And then I went to graduate school and I studied messenger RNA again, but this time in E. coli and bacteria. And then as a postdoc, and when I started my own lab, continued to study messenger RNA in the context of human diseases, and was able to show that human diseases can be due to mutations in DNA that cause misprocessing of RNA, or different kinds of mutations. And this is the work that I did at Hadassah Hospital with Eliezer Rachmalevich. Different kinds of mutations in our DNA can cause the mRNA to be degraded. So it was formed normally and then degraded. And so I never questioned whether RNA was important or not. I followed my nose, and every time I looked at something, it seemed to go to RNA.
Wow. And what about you, Adrian?
Yeah, well, DNA has this beautiful, iconic double helix structure, and RNA is just the wilder molecule, right? So it folds, and no two molecules look the same. And by the time I was starting graduate school, I was hearing all kinds of, or reading about all kinds of interesting things, like Tom Check finding that some RNAs could act as enzymes, ribozymes. So it just seemed like a wilder frontier, if you will. RNA can do many different things, and exists in many different forms. And it just made it all that more exciting and interesting. I knew that there were a lot of unanswered questions, and that's what kind of piqued my curiosity.
All right.
So was your graduate lab based on your interest in RNA?
Yes.
Yeah, I think it was broader. It was more based on gene expression. But for me, if you study gene expression, you can either study transcription or what follows. And so I ended up being more interested in what happens right after, because there's lots of steps, lots of points of regulation. And so that's what and in Tom Maniatis' lab, people studied many different areas. And so there was a lot of flexibility in what you could choose to study.
So now I have a question which takes us a little bit to the history of the RNA world. And what do you think was the turning point starting scientists appreciate RNA regulated process? What was the moment in the RNA field that made people change about their ideas about the importance of RNA? I would like to hear you, Joan. What do you think?
I'm not really sure of an answer to that question. At the time that I went to graduate school, we knew of three kinds of RNA: ribosomal RNA, tRNA, and messenger RNA. And it wasn't until after that that we began appreciating the other kinds of RNA and the roles they might play in regulation, and how ubiquitous it is and how pervasive. And I think that certainly, I became convinced that RNA was a very, very important molecule in cells quite early on. But I'd be interested to hear what others think turning points were.
Okay, so Lynne, what do you think?
Well, I agree with Joan. I think it's been a process. I don't think there was any Eureka moment in terms of scientists and then the general public becoming aware of the importance of RNA. I think scientists became more aware when they realized that most of our DNA makes RNA, and that there are, as Joan said, lots of different kinds of RNA. And the way genes are regulated is often by regulating the RNA that comes from them. And that RNA can actually serve as a regulator. In fact, it can serve as an enzyme. It's involved in protein synthesis. Its enzymatic activity is involved in protein synthesis. And I think in terms of the general public being aware, I think some became aware through, for example, Adrian's work, where people realize that RNA can be targeted as a therapeutic. And I agree with the statement that despite the horrible pandemic that we're now in the midst of, hopefully coming out the other end, the general public at large became aware of the importance of RNA with the vaccine.
Okay, so what do you think, Adrian?
Yeah, I agree that it's been a process. I mean, so with every important new discovery in the RNA field, one just realizes it's the versatility of this molecule and its central role. I mean, I think one important, it's more conceptual, but the idea of the RNA world and the evidence that RNA probably preceded DNAs and proteins in pre-life or early forms of life. And so then as different steps are studied in detail, you realize that, yes, that's a point of regulation, where it's just polishing the beginning or the end of the molecule or the things in between or changing individual bases. You know, every step along the way, as you know, can be a rate limiting step in regulating gene expression. So it's a body of work. And so the more we study it, the more we realize its importance and its versatility.
So now I want to ask a question which you already alluded to several times about the RNA therapy. RNA therapy just started several years ago. Where do you think it will go? Delivery of RNA was always a problem. Do you think you were lucky, Adrian, with SMA? And it's still a challenge. How do you see that?
Yeah, of course, it's a challenge. And so when RNA interference was discovered, people thought that, okay, this is going to give rise to all sorts of therapies, and it probably will. It just takes time. And people said, okay, what's the limiting, what are the three most limiting aspects? And they would say delivery, delivery, delivery. And if you think about monoclonal antibodies, which are now a very routine type of therapy, fairly routine, it also took 20 years or more to kind of work out all the technical glitches and improvements that are necessary to make it more everyday. And so I think SMA was definitely a good choice for a number of reasons, but that means that, for instance, in the nervous system, it's possible to do this over and over, and there's a lot of promising therapies that are far along in clinical development. And I think everything in the end is a technical problem. It's maybe harder to get to certain organs and tissues, but there are ways to work that out. And I think the mRNA vaccines are showing us that. I mean, I think at first everybody was very skeptical. Messenger RNA is just so unstable. How can you really deliver this as a therapy? And so they worked out ways. And I think using it as a vaccine is kind of an ideal scenario, because you don't need the RNA to hang around for a long time. It just needs to be there long enough to make a protein that functions as an antigen, and then you get an immune response.
So really jumping to the mRNA vaccine, is it going to be a solution for other infectious diseases or cancer? Could you see cases where it will not work? What do you think, Joan?
Well, I think it's certainly going to revolutionize vaccination, as we've already seen, that because of the technology, the first companies were able to go from the sequence of the virus to trials of the efficacy of the vaccine within just months, which is absolutely remarkable. And the reason is that you just plug in a different sequence. It's not a new technology. It doesn't have any glitches because you've introduced something new. All you've done is stick in a new sequence. So I think that certainly there, it's going to be absolutely revolutionary in terms of producing vaccines against all sorts of pathogens. Things like cancer are going to take a longer time because, cancer is not one disease. It's many, many, many different diseases. And they're going to have to be, if a vaccine approach were to be useful, it would have to be many different vaccines. So that's going to take a bit longer, in my opinion.
Did you have ever imagined that the pseudouridine, the basic question you and even me are asking about pseudouridine, its role, would be so important. And
I never guessed. I mean, okay, we knew that pseudouridine sort of helped stabilize RNAs, but the fact that it would make the difference between your body being able to tell that this is a foreign RNA and they should try to make antibodies against it and get rid of it, as opposed to being something that can get inside of cells and make its product, is just mind-blowing. And yeah.
I definitely agree. Okay, I want to ask you, Lynne, what do you think is next in the RNA research? Where do we go next? What we are still missing?
Oh, well, despite all that we know about RNA, there's a lot of things that we don't understand about RNA. So I think RNA research has a very bright future. This is because there's lots of things we don't understand about how RNA is made and what it does functionally, given that there's so many different types. And I think, well, when I think about personalized medicine, I think about RNA, and Joan just mentioned this. So a lot of diseases are due to mutations. They're either genetic or acquired in our DNA. So if we look at the DNA, we can see what the mutations are and we can see what disease they cause as a phenotype, looking at the individual. But I think if we want to understand, in many cases, what the mutation does, we have to look at the RNA that's made from the gene that has the mutation. And the idea behind personalized medicine is that the treatment is to the individual that has a particular disease and not to the disease itself, because different individuals with the same disease can have different mutations and different causes, and therefore different metabolic processes that have gone awry with their RNA. So I think we're just beginning to appreciate what can be done by studying RNA in the context of personalized medicine.
And when you appreciate that 98% of that virtually all of the genome is transcribed, but only 2% of it is actually coding for our proteins, figuring out what all those other RNAs are doing is going to happen someplace along the way in order to solve all the problems that we're talking about. And that's a huge endeavor. So hopefully it'll get faster.
So, Joan, you think it's more to understand the diversity and the function of molecules we do not know about them yet? Do you envision discovering some other molecules we have not seen?
Well, certainly modifications we have not seen. I don't know about molecules that we haven't seen. Modifications that take RNA to particular places, for instance, which is very exciting right now.
Or, you know, we computationally have a lot of, they're called long non-coding RNAs, but it turns out some of those RNAs make little proteins, they're called peptides. And we're appreciating how not only they evolved, but what these little proteins do. So I think Joan's right, we know what RNAs exist, but maybe how they're processed into other molecules, or how they do something like encode a short protein we never thought they did, is just ripe for discovery.
Yeah.
What do you think, Adrian? What in your mind?
I think
where should we go?
I think we should go where our observations take us. I mean, who could have predicted the existence of, I don't know, CRISPR-Cas9, guide RNAs, pen editing, other processes, so anybody tomorrow could be investigating some observations in some exotic context and discover a new phenomenon. Then this can be generalized or it can be applied. I think, when people early in their careers tend to think that all the interesting things have been done already. And I think those of us who've been around longer know that that's not true, and we can always be surprised. It's always been fun to go to RNA Society meetings or Cold Spring Harbor meetings and find out what's new and exciting this year. And some years it is really mind-blowing what has been discovered. And that sort of triggers all kinds of new activities. And so we will never stop being surprised by RNA.
Yeah, you mentioned that. I still remember the Cold Spring Harbor meeting where Laureate was presented.
Ah, yeah.
Yeah,
1984. Yeah, I think that's where I met all of
you. It was on the cover of the Cold Spring Harbor.
The abstract.
Yeah. And this booklet of RNA was this size, and now it's this size. So this is what I showed the students in my first class of the RNA world, how it was in the 80s and now how it is now. Adrian, what do you think about RNA therapy? Where should we go? What diseases, how do you see that? Is the SMA approach applicable for other genetic diseases? I mean,
yeah.
What is your dream about it?
Right. Well, so in SMA, so what we're looking there at RNA as a target, and we're using a synthetic nucleic acid that's a little bit like RNA, but a short snippet of nucleic acid to target that RNA and modify the way in which it's normally handled by the cell. And I think there's a lot of things we could do with that approach. You can change splicing, you can change stability. So ultimately you can change what the gene is producing. And so as long as you have enough information about the target and the disease, I think there's a potential to intervene. I mean, of course, there are challenges. Sometimes, yeah, Lynne mentioned personalized medicine. One can, and it has been done, to develop one of these antisense oligonucleotides to fix a defect that only occurs in one patient, nobody else. So that's the extreme of personalized medicine. But the challenge is, how do you cannot do a clinical trial in that case, or in maybe less extreme cases, there may be a few dozen or a few hundred patients, but there are technical challenges to bring this into a practical drug that can help people. I think these are challenges that are solvable, and it takes collaboration between academia and industry and society, right? Governmental agencies and so on. And so we're kind of learning how to navigate that and to make it a more efficient process. And I think with mRNA vaccines, it's like that too. When there is a challenge, then everybody finds a way to try to expedite and facilitate this because of the urgency. But I think similar models can be used to try to battle against cancer or other neurological diseases. I think we have tools that are very powerful. We just have to get better at exploiting them and bringing them to practice more efficiently.
So you see the future ahead of us to providing more and more surprises and help to medicine.
Absolutely.
Yeah. So now I want to sort of ask you a personal question. What did you feel when you were notified about this prize? Joan, please.
Well, the thing that really pleased me most is that Lynne and Adrian were also notified of this prize. And the reason I think that is that, you know, I feel that it's a prize that's going to the RNA field because of how we have all worked together in order to figure out things that are going on in RNA has benefited the actual production of a really good vaccine. And I just find that wonderful because, you know, way back when I started, I said, oh, RNA is very basic research. Someday it'll have impact on medical science. But I'm not sure I really believe that I would live to see that day. And it's just very, very satisfying to see what's happened and to celebrate it with two of my very, very highly respected colleagues.
Thanks, Joan. That's nice. And what about you, Lynne?
I was ecstatic, and I feel very privileged and honored to receive this with Joan and Adrian, who I've known and respected forever. And it's very nice. There's lots of people who work on RNA. There are lots of people who work on RNA and are doing wonderful things. I'm very happy that RNA is being recognized as something that's important and has a very bright future and is relevant to therapeutics and human disease.
And what about you, Adrian? How did you feel?
Yeah, well, same. It was a completely unexpected call, so a great surprise, great honor, and immediately they said that this would be shared with Joan and Lynne, and so I was also very happy. I've known both of them. We've been colleagues and friends for over 30 years, and so it makes the field look good. And the other thing is that I remembered that a former colleague of mine, so Barbara McClintock, who worked at Cold Spring Harbor, and so I overlapped with her by maybe 2 or 3 years. But she's an icon in biology, and she was a pioneer in every way. So this was the first award that she won, and eventually she won the Nobel Prize. But I mean, work that she had done much earlier, and so the Wolf Prize was the first to kind of recognize, her amazing contributions. So that was it was amazing to receive a prize that she had once gotten. So that was a special feeling as well.
Yeah, I still remember this time when I was just an undergraduate at that time, and I remember the time when she got this Wolf Prize. Yes, indeed, many people who got the Wolf Prize also got the Nobel Prize. So there is also hope for that in the future. So interestingly, when I asked you about the future, you talked mostly about the translational aspect of RNA. But what do you think is still missing from our basic understanding of the RNA world? Joan?
Well, there's a lot we don't understand about RNA structure. And Francis Crick always used to say, if you can't study function, study structure. And structure is, I think, revealing more and more about possible functional implications of RNA. So I think that's something that needs to be done even more vigorously than it's being done at present.
What about tools?
Oh, the tools that come along are absolutely marvelous. I'm not a person or not a lab that we don't devise that many tools, but we certainly appreciate the tools that our colleagues come up with, which are very, very ingenious and allow you to ask things and get answers that just were inconceivable before they came up with a particular technology. Technology truly does drive biological research. That's clear.
What about you, Lynne? What do you think is still missing for our basic understanding of this world?
Well, there's this whole new field called epigenetics, and this applies to RNA. So yes, RNA is made and it does its thing, but what can also happen, and often does happen, is that conditionally the RNA is modified after it's made or while it's being made. So the nucleotides that compose it are modified with extra chemistry. And we're just beginning to understand what's the significance of this extra chemistry to how our genes are ultimately being expressed under particular conditions. We hardly know anything about this, but we know it's important.
What do you think, Adrian?
I think lots is missing. The details, we just need to understand the details because, I study RNA splicing for, over 3 decades. We've learned so much. It's amazing, both function and structure as well. But we don't understand. We can't look at a sequence and precisely predict where all the exons and introns are, the boundaries, or under what conditions some exon is going to be included or not. And so we just need to have a deeper understanding to really be able to feel that we know the system, we know the rules, we know how it works, we know the mechanism. And that's true for every other aspect of how RNA is processed. And also, I don't think there is that much of a division between the basic and the translational research. They depend on each other, lots of, in the course of doing translational research, you can discover new things. Same thing goes for technology development. All these things have to go hand in hand. And that's part of what's very satisfying about working in this area.
So my last question to you, what would you suggest the young generation of young scientists, many are watching us, Joan, what do you would suggest for young scientists this time?
Well, I recommend RNA as a field that has come up with infinite surprises. Every place you look, there's something new. And I mean, one thing that's very exciting about doing science is that, as Adrian mentioned earlier, you expect a certain result and you might see that result, but then you see something else and you say, what is that? And invariably following that up leads to these Eureka moments and aha experiences. And so just in terms of doing the sance, if you enjoy doing this, then it's something you should pursue. And whether it's more applied or whether it's more molecular or whether it's more genetic doesn't really make a difference, because everybody has their own sort of level of satisfaction with the type of answers that you're getting, and they can be on these different levels. So I just say to young people, you know, go out there, try it, and if you like it, keep at it, and it'll be wonderful.
Thank you. And what about you, Lynne?
Well, speaking for myself, I got interested in what I got interested in because of what I learned as an undergraduate. And partly that was because of the science, and partly that was because of the lab environment. So we spent a lot of time in the lab, and it matters who we work with and what the dynamics in the lab are. But in the end, what makes scientists successful is their own individual perception of what to do, what experiments to do, and how to interpret them. So while I think RNA is a wonderful field, and I just feel so fortunate to have worked on it, I would encourage young people to work on something that they're inspired to do. And my advice would be, no matter what you do, it's really important to know what you know and what you don't know. It's really important to know what's factual and what's interpretation. And to understand when you interpret results, that there are going to be limitations, so don't overinterpret. And then if you follow your nose, I think you'll do well no matter what. It has to be, it's not always fun, but mostly it's fun. And if you like solving puzzles, science, and there's lots to do in RNA, are great ways to go.
Thank you, Lynne. And what about you, Adrian?
I agree with all of that. And I think that, if you're passionate about something, you're going to do well. I think I mentioned before, if you don't feel that all the interesting things have been discovered already, this is definitely not true. So, and don't be afraid of trying difficult things. You have to be persistent, but if you only go for the easy experiments or easy projects, then it's probably not going to be as unique or original, because other people will be trying that as well. Just don't be afraid. When you're young and starting out, you can take risks, and if you're persistent, the reward can be great, the satisfaction. It'll be like solving amazing puzzles, like Lynne said. So there's plenty to be done.
So I think our time is coming to an end, and I want again to congratulate you for the Wolf Prize. It was lovely talking to you. I enjoyed it. I hope you enjoyed it too, and so is the audience that followed us. And as I said at the beginning, we are in a special week this week in Israel. Tomorrow is our election, and on Saturday we celebrate Passover. So I wish you all again, lots of Eureka moments in life, and it was lovely talking to you. Thank you so much.
Thank you, Shula.
Thank you, Shula. You did a great job.
Thank you very much. Bye-bye. Shalom.
Bye-bye.
Bye. Shalom.
Bye. Shalom.